Hello everyone, I'm Alejandro Montenegro and I'm an executive editor at Cold Spring Harbor Protocols. I'm joined today by Carlos Riberio, who is a senior group leader at the Champalimaud Foundation in Portugal, to talk a little bit about the research and the work they will present at the 88th Cold Spring Harbor Symposium, which this year focuses on brain-body physiology. Thank you for taking the time.
It's a pleasure to be here.
So the work in your lab is centered around decision making. Particularly on how animals decide what to eat and how these decisions affect fitness in animals. I was wondering if you could tell us a little bit about that.
Yeah, so this started from the time of my postdoc. So I was very lucky to be in Barry Dixon's lab as a postdoc when the lab was transitioning from an action guidance lab to really start using molecular cellular tools to dissect behavioral circuits which generate behavior in Drosophila. And I was always a skeptical. I'm a trained cell biologist, developmental biologist, but then I realized, right, that actually we are getting there. We are getting there that we can start understanding really complex behavioral tasks at the cellular molecular level. And so thinking about what could be the kind of ethologically relevant task which Drosophila could do in terms of like value-based decision making, right? So choosing between different options in a value-based way where it changes actually what it chooses to do depending on its needs. Mm-hmm. Reading old literature, I discovered this fascinating world of nutrient-specific appetites. And so, yeah, I still remember the day I did the experiment, right? I compared flies, female flies, which had been protein deprived, as been described in classic insect physiology literature, with flies which have been kept on full diet and given the choice between protein and sugar. And I will never forget, right? I mean, all the flies which had been protein deprived had eaten yeast, which is a protein source, and I had given some blue color, so they were all blue. And the other flies, they actually kept on preferring sugar, which I had mixed with red color. And so that launched all of this. And for me, that's been a wonderful adventure because it's not only about decision making, but this opened up the whole world of nutrients, what's the impact of nutrients, understanding physiology, the interaction between the body and the brain, the gut microbiome, etc. So it really opened what I think is more of a research platform, which is the interaction between nutrients and the brain and how the body and the brain keep homeostasis. And that's just been a wonderful interdisciplinary voyage, which is what I like actually about our work.
So you mentioned that to address many of these questions, you use the fruit fly, Drosophila melanogaster. So what advantages does the fly feature that makes it a good model to study decision making? Well,
I think every model has its strength, right? And I think to a certain extent, I think also it depends also on what is the level of answer, what satisfies you as a scientist as an answer, right? And for me, the level I want to understand biological processes is first of all at the kind of interface of molecular and cellular level. And the other thing is at the whole animal level. And I think that's really where Drosophila shines, right? Taking something, dissecting it at the molecular cellular level, but doing that in the context of the whole organism. I mean, the classic work, for example, of developmental biology is a whole organism type of question, right? And I think flies lend themselves amazingly well also to questions of physiology and behavior because you need to query them in the whole animal context. And you can do that with extreme molecular and cellular stringency. And on top of that, you can do that in a hypothesis-free way, in a sense of like you can do screens. So you don't have to go in with a predefined hypothesis you test, but you can have actually the organism, the system tell you how it really works. That's why it's really one of the most amazing discovery systems you can use.
Mm-hmm. So research has shown that what animals eat affects not only their ability to reproduce, but also things like mood and mental state. Can these things be studied in a fly?
Absolutely, right. I mean, that's exactly the core of all our work, right? So as I said, I mean, the key experiment for me was the one where I deprived the animal of protein and then saw that now these animals all ate protein compared to the basal preference, which is to eat sugar. And that tells you that the brain has access to the nutrient state of the animal. and that it obviously then will change the preferences of the animal so that it seeks out and then takes in the nutrient which is essential, because that's the beauty also of nutrition, is that it's very conserved. So the same 10 essential amino acids are also essential in Drosophila. And so I believe, and I got trained, right? I mean, again, I'm a classic developmental biologist, trained in Biozentrum in Basel, right? Hox genes, etc. That you need to go in and ask very basic, very important questions which are likely to be conserved, and then actually look at them and how they're implemented in the animal. And so I think that the origin of many cognitive decisions and many cognitive processes which are about how to deal with resources, right? How to manage the trade-off of resources, how to remember where you get the right resource. All of these have as its origin basic needs like nutrition and obviously reproduction. And also we haven't evolved to deal with money as a resource, right? We use it as a proxy for other things which are, for example, nutrients. And so I think we can understand a lot and learn a lot about the molecular cellular principles which guide the impact of nutrients and the microbiome and the gut-brain interaction using Drosophila, and then look, especially at the molecular level, how they are conserved across phyla, including in humans.
Well, you mentioned that your questions are very multidisciplinary, involving behavior, metabolism, microbiome, and physiology. And I was wondering if you could tell us a little bit about such multidisciplinary approach and why you think that's the best way to tackle the specific problem of decision-making in nutrition.
Well, I think, again, it's the question of what is it you want to understand and what's the level you want to understand it. And I think that for me, one thing which is great about neuroscience or about developmental biology again and so on is that it puts a question at the center, right? Which is how do animals develop, or how does the brain function? How does it generate behavior? And ultimately then, what's the method, what's the approach you use then to tackle that is obviously dependent on your know-how, right? The tools you have available, but also what's actually pertinent to use, right? And I think that asking these type of whole organism type of questions, which require an across-scale approach, both across-scale meaning different entities within the animal will work together, like the gut, the reproductive system, the brain. They will interchange information all the time, right? To kind of make decisions and guide processes at the whole animal level, but also across levels at the level of understanding. So really going from molecular to cellular to circuit to organs to whole animal interactions. It's daunting. It's a daunting problem, right? But I think that's the excitement of the moment, is we can start putting things together, right? Like people did that 50 years ago, right? Where they started putting back biochemical processes together, either in tubes or in cells. We can start doing that now, but at the level of organisms, and also asking questions which are now like effects and phenomena which now will scale up and actually emerge from the interactions of all these things. And that obviously needs the work at the interface of molecules, neurons, imaging, metabolomics, really bring this all together. And I think that's why it's so fantastic to do science nowadays, right? It's because we have the tools. We sometimes don't know really how to use them yet, right? How to use them to find answers. We have maps, they're amazing. We have cellular maps now. We have these atlases. We did the fly cell atlas with many colleagues. We now have the connectome of the fly brain, so we know how every neuron is connected to which one. And so that creates an amazing playground to start putting things together and really find a deep mechanistic understanding of how these complex processes work.
Mm-hmm. I mean, regarding methods, in your abstract, you mentioned that you combine imaging approaches and neurogenetics and connectomics and other approaches, behavioral analysis as well, to get this mechanistic understanding. So how does all that thing come together to tackle a particular question? That's
a great question. So the way we kind of ended up coalescing, right? I could claim I designed this from the principle, right? But the thing which ended up coalescing is really building up around phenotypes, okay? And so the main phenotype we use in the lab is really this behavioral decision. What does the animal decide what to eat, okay? And so this started with this very simple red-blue type of assay, which I mentioned. But nowadays we are asking these questions using quantitative highly dynamic behavioral, using methods which allow us to quantitatively, automatically, and dynamically look at the behavior. So we use methods like the fly pad, a method we developed to measure how the fly eats from food, but also tracking, where we really look at the animal making decisions, foraging, looking for food, deciding how long to stay on the food or to look for something better out there, right? And these type of more proctocognitive decisions, that's really what we use. Does the manipulation of a neuron, of a genetic element, of a microbiome, bacterium, do they change this behavior? And if we see that there is a phenotype, then we dissect it using different methods, be it imaging, be it single cell sequencing, be it metabolomics, etc., with the idea to always then go back and functionally test if it has a functional relevance, which is again something you can do in flies, right? And I think that's what unites everybody, the biochemists, the metabolomics people, the people who do circuit imaging and analysis, is can we coalesce at the end back to the behavior, and then even go one step further and break the behavior and look if there is a fitness effect, right? Is there a reproductive effect? Is there an effect on longevity? Is that the reason why this trait was selected for? And that's what I find fascinating, is that we can go back and use all these different levels of understanding to get then a multifaceted, integrated kind of answer, what we call a mechanism, right? To kind of get insights into these complex questions like behavior or reproduction or metabolic homeostasis.
And is all that expertise within the lab, or do you collaborate a lot?
Well, that's another great question, right? I think science is collaborative, right? I think this idea of the lonely genius, right? I mean, that's not really how it works. And I think the way also I look at the lab is really as an ecosystem of people who bring in their own ideas and their own expertise. For example, we have a microbial geneticist who works in our lab. We have people who come from connectomics. But we also, we just recruited someone who has done a lot of hippocampal imaging and recordings in mice, right? And so we can learn then from these people and bring in their perspective. If there is a method which is really core to what we do, like calcium imaging, or connectomics or metabolomics, analysis especially, we try to establish that in the lab, or we are very lucky at Champalimaud. I work in one of the premier places for doing circuit neuroscience, so we collaborate also a lot, obviously, with people in-house. We also nowadays do zebrafish with my colleague Michael Orger, or we do mouse feeding behavior with Albino Maya in the Institute, but this has a limit, right? And so, for example, the connectomics is a highly collaborative work with many, many people there, mainly at Genelia, but also in Cambridge. Metabolomics, we worked with people in Glasgow. Now we have a new collaboration with Theodor Alexandrov, trying to establish spatial metabolomics in the fly. So when it comes really to bring in cutting-edge work, or for example, also at Champalimaud we don't have genomics, so we work with Bart de Planck or with Stein Ertz or with the Fly Cell Atlas Consortium. We collaborate with these people and we bring in our expertise because we know the animal really, really well, right? So we know which cells express with genes because we work across levels, so we know not only how the neurons look like, but also what we think they should be expressed in genes. But they bring in the technical expertise. And obviously, funders also need to work with us, right? And so we are very lucky to be getting funding from the Champalimaud Foundation, but also other funders which really allow us to push our work to new limits, like the Chan Zuckerberg, right? The La Caixa Foundation in Spain, and they all come together to allow us to really do this type of work. And collaboration is not the future, it's the now, right? It's just going to be more and more and more.
So what's next in your research? What are some of the questions that you're tackling next?
Well, you should ask the fly, right? Because whatever the fly tells us is where we go. Well, again, I'm currently very interested in moving into thinking about decision making in a more cognitive level, more abstract level. So beyond eating or not eating, but really strategizing, right? Should I stay on this type of food, which I just found, or should I go and look for something better out there? How do I trade off these type of cognitive decisions? And we find fascinating behaviors there. And also we're trying to understand how does the brain not only integrate internal nutrient and microbiome information, for example, but also the history of what's around it, the density of resources, etc. And again, also we are very interested in thinking about making these multimodal information, bringing that to the spatial level, right? So we are very excited to be working with Theo Alexandrov on spatial metabolomics. A lot of challenges there, but bringing that into the animal is something I'm excited about. And then also trying to think about across evolution, right? How much is it what we discover in flies? How much does it also work in other systems, including the human? So, you know, I'm not worried that we will get bored. And anyway, the things which will have the most impact and which I'm most excited about, I don't even know what that's going to be. Like the microbiome, it's not like we decided to work on the microbiome. It was the insight that sometimes flies would do the decision in one way and sometimes in the other way, which led us to see that actually there were differences in the microbiome, and that then launched us into that. So I think that's the beauty of discovery science, is that you don't know where you're going to go, what the next discovery is going to be, but as long as you put into the context of it being relevant for the animal, that's going to be interesting. And I think the other exciting thing is that we can now use all these tools to really find how it works in an animal. And the other thing I find exciting about working in Drosophila is that, you know, I can imagine in my lifetime to still see and really understand how all these molecular processes work together in neurons, which then work together to create these amazing behavioral outputs and physiological outputs. And I think if there's a system where we're going to crack this, it's going to be the fly.
Well, unfortunately, that's all the time we have today. I want to thank Carlos again for being with us, and all of you for listening.
Thank you very much.